Like nuclei, mitochondria are surrounded by two membranes, an inner one and an outer one. It is, of course, not perforated. There are no mitochondrial membrane pores. The cartoon emphasizes the double membrane and the familiar elaboration of the inner membrane into cristae, which can also be seen in the transmission electron micrograph. Look at the cell fractionation of mitochondria in TEM on this slide. Actually, these structures don't look a whole lot like what you would see in situ in cross-sections of cells, but they are about the same dimensions as mitochondria. Knowing the respiratory function of mitochondria, what would you do to confirm that your fractions are indeed mitochondria? Here are some experimental tests. You could check to see if the isolated structures would oxidize pyruvate or other substrates. What other substrates might you test to see if they're oxidized by this cell fraction? You could check to see if the isolate would convert ADP and inorganic phosphate into ATP, another function of mitochondria. What are some other experimental tests that you could do to prove that your isolated cell fraction are indeed mitochondria?